Hello everyone, I'm Anas Al Ubaidi, and this is session 35 or lecture 35 within chapter 24 of the Advanced Medical Science Liaison and Medical Monitor Certification course program by CCRPS. The chapter is about conceptual and theoretical monitoring, while the topic for the session is about monitoring of drug context and drug environment interaction trials. Introduction. We'll start with the definitions with an introduction. As for drug context interaction, trials assess how a drug's effects are influenced by situational factors such as the setting in which it is administered or the psychological state of the patient. While on the other hand, for the drug environment interaction, trials explore how external environment factors such as climate, socioeconomic conditions, or lifestyle affect a drug's absorption, efficacy, and safety. 2 points. We'll start with the patient safety or the participant safety. As the focus here, the primary goal is to identify risks posed by environmental or situational factors that could compromise patient health or drug efficacy. And here's a summarized diagram on an unveiling patient safety dimensions as on the left patient safety, on the right ADR monitoring, simulated trials and environmental factors. Actions: adverse drug reaction (ADR) monitoring, track how drugs interact with environmental triggers to prevent harmful outcomes, as for example, a blood pressure medication was tested on patients living at high altitudes. Adverse reactions like increased hypertensive episodes were linked to oxygen deprivation, which required adaptive dose modifications. Simulated trials under environmental variables by recreating specific environmental conditions, researchers preemptively detect risks, as for example, a chemotherapy drug trial exposed patients to simulated hot climates, revealing nausea as an unexpected side effect under heat stress conditions. Data integrity. The focus for data integrity. Accurate and consistent data is essential for comparing drug performance across variable settings and environments. And here's a summarized diagram on balancing data integrity in drug trials. On the left, we have controlled environments, consistent data protocols. On the right, real-world environments, variable patient outcomes. The actions here for data integrity: standardized data collection procedures ensure unified protocols for recording data at multi-location trial sites. Example, a diabetes medication trial validated outcomes by ensuring consistent blood glucose measurement techniques across urban and rural testing sites. Controlled versus real-world comparisons analyze discrepancies between results in laboratory conditions versus real-world environments to identify variability. Example, research showed that sedentary urban patients had poorer glucose control compared to their active rural counterparts when on the same anti-diabetic medications or medication. Protocol adherence. The focus for protocol adherence: consistency in protocols eliminates biases or errors stemming from differences in testing environments. Here's a summarized diagram on ensuring protocol adherence in trials. Left to right, identify focus, establishing the need for consistency in protocol, standardized parameters, setting uniform environmental conditions across sites. Example implementation: applying controlled environments in a drug trial, conduct adherence checks, monitoring compliance through regular inspections. Actions for protocol adherence: controlled environmental parameters, standardized variables such as room temperature, lighting, and testing equipment across all trial sites. As for example, a cognitive enhancement drug trial ensure participants perform tasks in identical controlled environments to neutralize disruptions affecting outcomes. Adherence checks regularly monitor sites to ensure compliance. Regulatory compliance. The focus for regulatory compliance. Trials in diverse contexts and environments must meet stringent legal and ethical standards to satisfy approval agencies like the FDA in USA or EMA in EU. Now here a summarized diagram on ensuring compliance in drug trials. As within regulatory compliance, uh, bottom to top to bottom, we have environmental influences, dietary habits affecting drug efficacy, working conditions impacting trial results maintaining standards in adverse conditions, safeguarding vulnerable populations, and then ethical oversight, all towards meeting regulatory standards for drug trials. Continuing for within the actions for regulatory compliance, documenting environmental influences, transparent reporting of how drugs respond to regional dietary habits or working conditions aids regulators. Example, a chemotherapy trial revealed higher nausea in hot climate populations, prompting specific labeling requirements for regions with extreme weather. Ethical oversight maintain ethical standards, especially when testing drugs in vulnerable populations or adverse environmental conditions. Steps to effective monitoring. Step 1, selection. Choose geographically and environmentally diverse trial sites to capture variability in responses, as for example, a malaria prophylaxis trial conducted comparative studies in tropical regions with high humidity and temperate regions, revealing variations in drug performance tied to temperature, extreme, extremes, or extremes, or extremes. Here is a summarized diagram on enhancing clinical trial monitoring. As here we have site selection, as we read. Here's in step 1. Then we have risk-based monitoring as step 2, RBM, and then data review as step 3. Step, Continuing with the site selection, ensure local expertise to manage environmental challenges. And here's step 2, RBM, risk-based monitoring. Allocate resources and prioritize populations exposed to greater environmental risks, as for example, patients in urban areas with high pollution levels may require additional assessments in lipid-lowering drug trials as pollution impacts inflammation pathways. Step 3, data review. Use centralized analytics to identify environmental or situational trends in drug response, as for example, an antiviral drug trial highlighted reduced efficacy in shift workers due to disrupted sleep cycles, prompting additional trial designs to explore time-dependent dosing. Tools and techniques for monitoring. We have the GIS, Geographic Information Systems, purpose correlate drug responses with environmental variables like climate or pollution. Example, as here, GIS mapping showed increased joint pain for arthritis patients in colder regions, influencing dosage recommendations. Remote patient monitoring, RPM, purpose real-time collection of patient data from diverse settings using wearable devices, as for example, a heart failure drug trial employed RPM, remote patient monitoring devices to track cardiac responses across hot and temperate climates. Here's a summarized diagram on visualizing monitoring techniques in healthcare, as on the left, monitoring techniques, on the right, geographic information systems, GIS, remote patient monitoring, RPM. Continuing with the tools and techniques for monitoring, centralized data management systems purpose aggregate data from multiple sites while ensuring consistency in collection methods. Example, a clinical trial assist the uniformity of data between under-resourced rural clinics and urban hospitals using centralized cloud databases. Environmental simulation technology purpose here recreate external conditions to model drug responses in controlled labs, as for example, a hypobaric chamber simulated high altitude oxygen levels for monitoring anemia drug effectiveness. Examples of successful monitoring, example 1, a trial here, asthma indication is polluted urban areas, outcome adjusted dosing schedules to counter diminished efficacy in high pollution environments. Here's a diagram summarizing the successful clinical trial monitoring asthma trial urban areas, left to right. Analgesic trial for athletes, insomnia drug in shift work, and anti-diabetic drug in urban and rural/rural. Example 2 trial analgesic tested in high-altitude training athletes, outcome altered drug absorption patterns led to refined athletic use guidelines. Example 3 trial insomnia drug under simulated shift work conditions, outcome verified strategies for dose timing to enhance sleep without impacting functionality. Example 4, as for trial anti-diabetic drug tested across urban versus rural lifestyles, outcome personalized recommendations using activity adjustments for stronger outcomes. Summary tables. Next, we have tables to summarize what we have covered through this lecture. We have table here on key differences between drug contest and drug environment interaction trials. Aspect comparison in between both for each aspect, as for definition, examples, scenario focus, trial scope, and personalization impact. Table on key components of effective monitoring, component focus example, components we have patient safety, data integrity, protocol adherence, regulatory compliance, personalization factors. Table on steps for effective monitoring in diverse conditions, step criteria or action examples, steps we have site selection, risk-based monitoring, data review, centralized data management, environmental simulation. Table on tools and techniques for monitoring, tool or technique purpose example, tools or techniques we have geographic information systems, GIS, remote patient monitoring, RPM, centralized data systems, environmental simulation, wearable devices. Table on examples of successful monitoring applications, trial, environmental or contextual focus outcome trials. We have asthma medication, analgesic for athletes, anemia drug, diabetes, metabolism, hypertension treatment. Application table, scenario for each scenario, key principle applied, key actions, timeline, what to watch out for. As for scenarios, we have monitoring blood pressure, medication effects in high altitude conditions, tracking urban versus rural diabetic drug outcomes, examining anti-anxiety drug efficacy in an emergency room, high stress versus home, low stress settings, evaluating arthritis medication effects during colder seasons in trial regions, assessing chemotherapy drug nausea rates in hot climate zones, analyzing cardiovascular drug responses near industrial high pollution zones, testing antiviral drug efficacy among shift workers versus regular scheduled patients, studying anemia drug absorption in night shift workers with limited daylight exposure. We have tailoring diabetes drug regimens for high carb diet populations, high altitude simulation for evaluating oxygen effects on a painkiller, remote monitoring outcomes for cardiac patients during temperature related stress, comparative nausea documentation across diverse food patterns and multinational chemotherapy trials. Next, we will revise what we have covered through this lecture through some multiple choice questions or MCQs. Question 1. What do drug context interaction trials primarily evaluate? A, the financial implications of administering a drug in hospitals. B, how situational factors such as psychological state influence drug effects. C, the long-term environmental impact of drug manufacturing, or D, how drugs interact with different enzyme systems globally. So definitely the answer is B, as of how situational factors such as psychological state influence drug effects, as the drug context interaction trials focus on how situational factors like the setting or psychological condition alter a drug's efficacy, safety, and adherence, ensuring realistic understanding of drug performance. Next question, which tool is most suitable for linking drug responses to geographical and environmental conditions? Is it A, geographic information systems, GIS, B, centralized prescription logs, C, heart rate variability monitors, or D, standard physical health tests? Tests. Definitely the answer is A, as how to link drug response to geographical and environmental conditions, as it is A, geographic information systems, GIS, as geographic information systems, GIS help identify patterns by mapping drug responses to specific environmental or geographic factors such as climate changes or regional specific risks. Question 3. What is the primary focus of ensuring protocol adherence in drug context trials? Is it A, evaluating cost effective of drugs distribution, B, maintain consistency in testing conditions across varying environments, C, comparing city-based versus rural health outcomes, or D, reducing levels of patient stress during administration? So primary focus, what is the primary focus of ensuring protocol adherence in drug context trials? Definitely the answer is B, as maintaining consistency in testing conditions across varying environments. As protocol adherence ensures uniformity in environmental and procedural conditions during trials, eliminating biases caused by factors like lighting or temperature variances. Question 4, how do remote patient monitoring RPM tools assist in these trials? A, by automating drug composition testing in laboratories. B, by providing real-time environmental data for patient health metrics. C, by reducing trial costs through fewer follow-ups. Or D, by replacing laboratory testing with patient-reported symptoms. With patient reporting symptoms. So how do remote patient monitoring RPM tools assist in these trials? The real-time data. So answer is B, by providing real-time environmental data for patient health metrics. As RPM tools that capture real-time physiological data, such as cardiac performance and the various environmental conditions, making them vital for monitoring diverse real-world scenarios. Question 5. What was significant finding from trials evaluating asthma medication in polluted cities? A. Pollution has no notable effect on asthma drug efficacy. B. High pollution decreased asthma medication effectiveness, prompting adjusted dosages. C. Asthma medications are more effective in polluted environment. Or D. Urban patients have had higher overall adherence rates due to better awareness. So we know for such asthma, as for example, for evaluating asthma medication in polluted cities, are for such trial which came up during the lecture, as for example, we know the outcome and we recognize as the outcome was medication adjustment due to the effect of the pollution on the effectiveness and the efficiency of the drug. So the answer would be B, as high pollution decreased asthma medication effectiveness, prompting adjusted dosages. As trials found that high pollution diminished asthma medication efficacy, highlighting the need for tailored guidance and dosage adjustments for patients in polluted settings. And here we come to the end of this lecture. Thank you very much and goodbye.